Match the following terms to the definitions. We're going to begin with conjugation. It is a genetic material. It's transferred from one bacterium to another. It's from the donor cell to the recipient cell using pili. Transcription. It is the synthesis of a complementary strand of RNA from a DNA template. Transduction. It's bacterial DNA is transferred from a donor cell to a recipient cell inside a virus that infects, let's continue here, that infects bacteria. Transformation, D. That is, genes are transferred from one bacterium to another as, quote, naked, end quote, DNA in a solution. And then translation, E. It is protein synthesis. All right, we have our definitions for our terms. Let's answer our first question. Transfer of DNA from a donor to a recipient cell by a bacteriophage. A bacteriophage is a virus. Now, which one of the definitions has a virus in it? Well, bacterial DNA is transferred from a donor cell to a recipient cell inside of a virus that infects bacteria. So our answer is C. It says all of these components. Next one, transfer of DNA from a donor to a recipient as a naked cell. Well, which one has the donor, recipient, and naked DNA in a solution? Well, that one has to be D. So we have, for the first one, C, it's transduction. And then the D is transformation for the second one. Make sure you write that one out, those out. All right, so we're done with those two. We're going to go to our next question, our multiple choice. Feedback inhibition differs from repression because feedback inhibition A is less precise, B is slower acting, C stops the action of pre-existing enzymes, D stops the synthesis of new enzymes, or E, all of the above. Let's define our terms. Feedback inhibition. It is the overproduction. So I'm just going to initial or just say it's FI. So it is the overproduction of the end product, which can inhibit an enzyme, Z, in that pathway. So we have feedback. What is repression? Repression is inhibition, or the inhibits. Let's go back. It inhibits the gene expression and decreases the synthesis of enzymes. We have our definitions for our terms. So feedback inhibition differs from repression because feedback inhibition is less precise. Well, that's not in our definition. Is slower acting? No. You can cross these out. Stops the action of pre-existing enzymes. Well, that's what it does. It inhibits an enzyme that's already existing in that pathway. So the answer is C. Our next question. Bacteria can acquire antibiotic resistance by all of the following except. So we want to know by process of elimination, bacteria can acquire bacteria can acquire antibiotic resistance. How can it acquire it? Well, it can acquire it through mutation, so that is true. B, insertion of transpons. Well, what is a transponson? Well, let's define that. It is small segments of DNA that can move from one region of DNA to another. We have conjugation. Well, since transponsons is a small segment of DNA that can move from one region of DNA molecule to another, that's a way that bacteria can acquire antibiotic resistance. So that is true. Conjugation, which we define up above. Let's go and check that one out. So we have conjugation. A genetic material is transferred from one bacterium to another. It is the donor transferring its genetic material to the recipient using pili. D, we have SNRPs, and that is small nuclear proteins, and they remove introns and splice. That looks so ugly. Splice exons together. Now, introns are intervening regions of DNA that do not encode protein, and exons are regions of DNA that are expressed. So that's SNRPs. And then transformation, which we defined above. Transformation, that's D. Genes are transferred from one bacterium to another as, quote, naked DNA in solution. So we have our definitions for our terms. And we want to know the bacteria can acquire antibiotic resistance. How can it? It can through conjugation, and it can through transformation. What's left is D. So the answer is SNRPs. Suppose you inoculate 3 flasks of minimal salt broth with E. coli. Flask A contains glucose. So we have A contains glucose. We have flask B that contains glucose and lactose. And then we have flask C that contains just lactose. After a few hours of incubation, you test the flask for presence of beta-galactosidase. So that is present. Let's put that right here. Present. And it's beta-gal. Which flask do you predict will have this enzyme? Let's see. What does beta-galactosidase do? Well, it is an enzyme that splits the substrate lactose into 2 simple sugars, glucose and galactose. Another helpful information is when glucose is present, when there's a high amount of glucose, then there's a low amount of CAMP, which reduces the lac operon activation, thus giving a low production of beta-galactosidase. So when glucose is present, you get a low amount of beta-galactosidase. So it cannot be A and it cannot be B. So we're going to cancel those. And it cannot be B. What we're left with is C. Because glucose is present in lactose is present in flask C, and when there is lactose present, then beta-galactosidase will be able to split it into simple sugars like glucose and galactose. So that's C. That's the answer. Plasmids differ from transponsons in that plasmids. Well, what is a plasmid? Plasmid is a self-replicating gene containing circular pieces of DNA. And a transponson is a small segments of DNA. All right, plasmids differ from transponsons in that A, because become inserted into chromosomes, are self-replicated outside the chromosome, move from chromosome to chromosome, or carry genes from antibiotic resistance, or none of the above. Well, we know that from our definition that a plasmid is self-replicating gene containing circular pieces of DNA. And we see that here in B are self-replicated outside the chromosome. So that would be our answer, B. Use the following choices: catabolite, repression, DNA polymerase, induction, repression, or translation. Let's define these. A. It is glucose inhibits lac operon, mediated, mediated. Let's make that clear. Oops. By Camp CRP complex. B, DNA polymerase. That is an enzyme that joins new nucleotides to growing DNA strands. Induction is a process that turns on transcription of a gene or genes. Repression inhibits gene expression and decreases the synthesis of enzymes. And translation is protein synthesis. All right, now that we have our terms, let's go to our first multiple choice. Our first multiple choice. Mechanism by which the presence of glucose inhibits the lac operon. Which one gives us the glucose inhibits the lac operon? Well, we see that here in A, glucose inhibits lac operon mediated by CAMP CRP complex, and that is a catabolite repression. So the answer is A. Catabolite repression. Next one, the mechanism by which lactose controls the lac operon. Which one controls the lac operon? Lactose controls the lac operon. The process that turns a transcription of gene or genes. The answer is C. And that is induction. Two offspring cells are most likely to inherit which one of the following from the parent cell? A change in a nucleotide in messenger RNA, a change in a nucleotide in tRNA, a change in a nucleotide in rRNA, a change in a nucleotide in DNA, and E, a change in a protein. Well, the answer is D, and the reason why the answer is D is that DNA is replicated and can be passed to their offspring. Which of the following is not a method of horizontal gene transfer? Let's define horizontal gene transfer. Now, it's a transfer of genes from bacteria to other bacteria in the same generation, versus a vertical gene transfer, which occurs when genes are passed from an organism to its offspring. So we want to know which one is not a method of horizontal gene transfer. Another way to look at this question is what are the ones that are have the method of horizontal gene transfer? Well, let's go down. Binary fission is prokaryote cells reproduce by division into two daughter cells. Conjugation. A genetic material is transferred from one bacterium to another. It is a cell-to-cell contact. It's from donor to recipient, which involves pili. We have integration of transponsin. Is a small segment of DNA that can move from one region of the DNA molecule to another region. Transduction is the bacterial DNA which is transferred from a donor cell to a recipient cell inside of a virus that's infected by a bacteria. And then our last one, transformation, is genes are transferred from one bacterium to another as, quote, naked DNA in solution. Now we see that DNA is transferred from one bacterium to another in transformation. So that's true. Transduction, bacterial DNA is transferred from one donor to another. So that is true. Transponsin, small segments of DNA that can move from one region of DNA to another. That is true. Conjugation is genetic material that is transferred from one bacterium to another through pili. All of those are horizontal gene transfers. A is the answer. One, it's the one that's left. And we know by definition that binary fission is the prokaryotes' cell reproduction by division into two daughter cells.